Okay, so continuing with the theme of thinking about genomes, you just got a really nice introduction to the processes involved with getting a hold of sequences and then assembling those sequences. So you've got a genome. Yes. Oh, and I've got a thing that I was supposed to say, but wait, it's in which? Random? So if you could please, at some point, maybe not exactly while I'm talking, unless you get bored with what I'm talking about, go to the random conversation or channel in the Slack system and fill out, and there's a link, I guess, to a survey that Michele has provided, which he's asked me to have you fill out. That would be great. It would be information that he would be grateful to receive. That was it? Yeah, okay. All right, so you've gotten this introduction to getting sequences and to assembling those sequences. So you've got genomes that are mostly assembled, and you've been exposed to the ideas about how to annotate what makes up those genomes, those assemblies, covering a lot of different types of things that you might annotate. And that's from a perspective of somebody who does a lot of this for a living. I'm going to try to give you a little bit of a perspective about this annotation part of the process from somebody who builds tools that relate to annotation across a pretty wide range of annotation processes. I'm not going to dig deep into the details of how that works, but if you find yourself excited about those methods, come and grab me afterwards, particularly those who are in the genomics subgroup are going to have an opportunity to grab more information from me as well. But hopefully this will give those of you who haven't seen much of this before some kind of an alternate exposure to these topics. There's going to be some redundancy with what Jeremy just covered because we didn't synchronize exactly, so apologies for any redundancy. Maybe it'll work. If I'm standing close, it works. Okay, so just to reiterate what sequence annotation is. Nope, it's really not going to go as quickly as I want it to. Okay, so maybe we're thinking about what we mean when we say to annotate something, right? If I'm trying to annotate a thing, it doesn't have to be sequences, but in the context of sequences, we're looking to identify things that are present in the thing I'm trying to annotate, identify that there's a thing there, and there's a thing there, and there's a thing there, and not just that a thing is there, but also what that thing is. Is it a protein coding region? Is it a functional RNA region? Is it a transposable element? What those things are, and then not just what it is, but maybe even get to the point that we can assess what its function is, what its role in the organism that we're trying to annotate is. And Jeremy's given a nice introduction to approaches that are taken to do that. Let's talk a little bit more about some of those issues. So examples, as Jeremy has covered, include, look, you can just press a button. If you've got an assembled genome, you might ask where are the protein coding genes, what's the function of those genes. Proteins are made up of domains, so where are and what are the domains that make up the protein. If you've got a eukaryote, so that there's splicing that pulls introns out of the transcripts, where are the exons and the exon-intron boundaries, where are the functional RNA, where are the regulatory regions, either upstream or maybe within the introns of those genes. What's all the stuff that isn't one of the above things? And Jeremy's nicely introduced the notion of transposable elements and tandem repeats, which constitute most of that other stuff. And what that ends up looking like, maybe I'll get lucky, oh good. What that ends up looking like in when you look at those, you could either look at a GFF file, that text file that Jeremy showed you earlier, which you usually don't want to spend much time looking at if you're trying to make much sense of things, because text is hard to read when there are billions or millions of things. Is that you can look at a thing called a genome browser, and this right here is a screenshot captured from the UC Santa Cruz Ucsc genome browser, which is one of a small number of comprehensive browsers that contain all of this annotation information that's already been captured for existing genomes, right? You may, if you've got your own new genome, you have to build this stuff yourself, but if it's an existing genome like the human genome, you might be able to go and take a look at a particular region of that genome. Here we're looking at part of chromosome, that's the wrong button, chromosome 4. This is a little bit under a megabase of sequence, 1 million nucleotides of that genome. And there's about a dozen genes, each one of which is listed with or shown, visualized with these little blobs of sequence. And not only are there and really close. Okay, not only are there genes, but within those are the exons, which are each of the vertical dots that are present in here. And you can zoom in and out of regions of this, of these genomes to get more information about them. Not represented here in the genome browser, but in other approaches that you can use when you're trying to visualize these things. You might be able to, for example, using the PFAM database, you could identify particular domains that are present along the sequence of a protein. And as Jeremy pointed out, right, those domains tend to be regions that fold up into some kind of a coherent whole. Here's an example of a pyruvate kinase, some kind of kinase, which is in my notes, and it's got 3 domains that you can see that each coherently fold up into a thing, and those are just sequential blocks of sequence that fold up nicely into some component, and you can get that information by using by searching for particular domains within the larger protein sequence. Those domains can be made up by 1 or more exons along the stretch of the gene. Not all of your genome is protein coding or functional RNA coding. In fact, the vast majority of it isn't. Maybe 2% of your genome or so encodes something that's recognized mostly as being functional that gets transcribed and acts based on that transcript. Most of it is the remnants of ancient copies of transposable elements, these selfish, self-replicating elements that are in your genome, or just repetitive sequence, the tandemly repetitive sequence, ATG, 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 where copies of that have expanded over time based on replication slippage during the process of replicating a genome. And you can see all of that in these genome browsers, again, represented in this way with these individual tracks that show these different sorts that Jeremy alluded to of both transposons or retrotransposons, and then these simple repeats also being tracked, where these are identified by various tools that include repeat mask error, but others as well. And I want to talk a little bit about then, great, so you can either start with your own genome and annotate it, as Jeremy's described. You can say, well, I don't need to do my own. I want to go look at the annotation that someone else has already figured out for a genome that I'm interested in. So you can look at a genome browser to grab that information and download GFF files that are associated with all those visualized bits of information. But you might be interested in how all that annotation happened in the first place. And you've seen some examples of how that's done. I tend to think of it as being done by these 3 sort of general phases or approaches to annotating sequence. Stuff that's model-based, talk about that in a minute. In particular, approaching this topic of interspersed repeat modeling or transposon modeling, and then masking, and then similarity-based annotation, which really makes up the lion's share of annotation that you might see. We talk a tiny bit about each of those, not going to get into too much depth about them. Model-based annotation boils down to having some kind of a general model of what a type of thing that you're trying to annotate looks like, right? And searching for those. So examples of that might be, as Jeremy pointed out, protein coding genes. Like, what do they look like? Well, they've got open reading frames, runs of nucleotides that could be encoded as codons without a stop codon that would cause a run of encoded amino acids to stop producing a viable protein sequence, right? They can't be particularly short or they wouldn't be interesting, so a relatively long run. They can be broken up by introns, so you want to think about, hey, I've got an open reading frame followed by something that follows that intron donor and receptor site signal and matches up with another open reading frame downstream. You don't need to know what the protein does. You can just say, oh, it's an open reading frame with the classic splicing acceptor site following it, and then the donor site afterwards. And that gets you pretty far towards recognizing something that might plausibly be a protein coding gene in a eukaryote. You don't even have to worry about introns if you're in bacteria. And then these other sort of promoter sequences and other signals that may be even more informative in surrounding context. If you are seeking RNA, functional RNA, although this can be a little bit of a challenge to do, if you see compensatory runs of sequences, you've got a run of sequence and then the reverse complement is pretty close by, and you think about, well, what would that look like if it happened to get transcribed? Is that those two pieces would bind together and create a stem, and if you see a lot of that going on, then you might be looking at an RNA gene, even if you don't know what it does. Right? So that could be a model that would get you in a de novo form some idea of what kind of sequence you're looking at. Tandem repeats, right? The same sequence unit repeated over and over again, possibly with some substitution or insertion or deletion mutation. Those are repetitive. So if you had some kind of a model of what repetitive sequence looks like, you could model those and find them without needing to compare them to other previously observed or known tandem repeats. Jeremy's pointed out that Augustus is one of the tools that can be used for identifying probably proteins, things that are probably proteins in a genomic sequence, even if you don't know what the protein is or does. GeneMark and ORFinder, and there are a variety of other tools that do that sort of a thing. And all of these tools can be aided by external information, such as what's done with PASA, where you can use RNA sequencing results that can inform you that, oh, it looks like this region is getting transcribed a lot. And if that's getting transcribed a lot, then it's probably, not necessarily, but probably a gene of some sort. And if it happens to also have a nice open reading frame, then it's probably a protein, for example. Methylation signals, DNA accessibility experiments can give other insights into things that might be encoding genes from the gene sequence without needing to necessarily know what they are. So those are all things that are just, you can think of them as de novo identification. You don't need to know other things that look like them. You just need to have a model of what the thing is that you're looking for. There's a lot of other sorts of models like that you can come at it and say, you know, you squint your eyes and say that looks like the kind of thing I'm interested in, and so you can label it as such. And that, depending on how deeply into the weeds you want to get with annotating or labeling a genome, you may find yourself building up a stack of such tools to try to identify those things. Okay. So that was model-based annotation in a super quick nutshell. Jeremy's alluded to this fact that there are these transposable elements, which, right, a single master copy may, at some point in the past, tens, hundreds of millions of years ago, have been active and made another copy somewhere else in the genome, or 10, or 100, or 1,000, or if it's an ALU, 1,000,000 copies of the same basic element with minor, very minor differences back when those things were dropped in. ALUs are specific to primates, but they make up about 10% of your genome, right? So these are a bunch of things that are only a few million years old and are 300 base pairs long, and in many cases, they're really similar to each other, and it's one of the things that poses such a problem when you're trying to perform assembly or even understand what the content of a genome is. But once you've got an assembly, which you hopefully have been able to pull off because you've got long reads or proximity ligation approaches to try to improve it, then you maybe just want to know what's there. right? And so you can use Repeat Masker as a piece of software that you can use. And what Repeat Masker does is actually, it falls under the next heading that I'm going to describe in just a minute, which is similarity-based approaches, because it'll take the sequence that you're looking at, and it'll compare it against a library of known things. And we'll talk more about that in a second. But if you're looking at a genome and you want to know what all of the repetitive content is, another thing you might want to do is just say, I've got my sequence, and let me take that sequence and, if you want to be really slow about it, compare it against itself and see if there are regions in the genome that look like other regions in the genome. And if you see a lot of places in the genome that all look really similar to each other, to say, well, that looks like it's probably something that's repetitive in some way. And repetitive in particular means if they're interspersed in that way, that it probably at some point in the past was the result of some kind of a retrotransposition event, some kind of a copying event that made many copies. And so those things that you see, those artifacts that you see, are the remnants of some kind of ancient copying activity. So we should document what those are. And the process of doing that, there are a variety of tools. I've got the two. Nope, I don't have them up yet. I think I will here. What I think are the two biggest tools in that realm, one of which is Repeat Modeler, another one of which is Repet. I work with the developers of Repeat Modeler pretty closely, and we've contributed some of the methods into that tool. And we're now working with the Repet developers as well. Repeat Scout is a little bit older, and it serves as one of a variety of tools that serve as the basis of both of these tools. And if you're interested in this kind of thing, if you ever find yourself trying to annotate a genome, there's a really nice new guide to help new TE annotators get a handle on exactly what kind of strategies they can use for labeling those transposable elements, these repeat things. There's a lot of tools that you can bring to bear, and this is, I think, the first really good tutorial on how to approach that problem if you aren't a TE expert already. I'm not going to belabor this point too much, but it's a really important part because your genome is, and the genomes of most of the organisms you're likely to interact with, if you're not doing bacteria, are like 60%, or yours is at least 60% TE remnants, right? So if you're talking about 60% of your genome, you want to be sure that you found what's repetitive, because that repetitive stuff can both interfere with gene annotation approaches, as Jeremy recommended, because there's a bunch of protein coding, ancient pseudogenized protein coding regions within there, right? Stuff that's no longer functional because it's got random mutations that have occurred, but they still look a lot like proteins, and that'll really screw up gene finding approaches. But also, it turns out that these transposable element copies in many cases have been neofunctionalized or subfunctionalized, and those proteins themselves might be serving roles that you'd like to know what they're doing and understand the relationship to other functions that TE genes engage in, or they can control regulation of those genes in ways that are specific to the ways that TE sequences do that. And so knowing what they are is a pretty important aspect of annotation. Okay, so the last of this, what I'm describing in these classes of annotation approaches that you might use, is the one that really is responsible for the vast majority of annotation that happens with most of these, most genomes, which is similarity-based annotation. And the idea behind that is you say, I've got some sequence, it's not annotated. I compare it to some collection of sequences that I know something about. There's a database of sequences, or sometimes called a library of sequences. And if the sequence that I've got in my hand looks like some sequence in that database or library, more, and by looks like, I mean it's more similar to that sequence than you would expect by chance, right? Then you can transfer information from the one to the other, right? You can use that to inform the annotation of this sequence that you have in hand. This is what Jeremy was doing, for example, with AAT, is that's a tool that is comparing the genome to a bunch of known protein sequences and if the sequence matches one of those known sequences effectively and allowing for intron splicing to occur, then it can say, well, this part of the genome is one of those genes, and this part is one of those genes, right? And so these library searching approaches or database searching approaches are really at the heart of most gene annotation that occurs. There are tons of caveats to how that's done, most of which I'm not going to get into. Simple examples of that are like, what if I've got sequencing error, which can be more or less of a problem depending on what sequencing technology you've used. What if the thing that I'm searching with matches not just one thing in that database, but a bunch of different things in that database? What kind of strategies will we use in order to decide what it is? And there are some techniques, if I'm annotating a genome, that aim to do a pretty good job of picking the winner, right? The winning annotation, if there are some competing annotations, but there's a lot of complexity to that often isn't addressed. If you're interested in those topics, grab me after and I'll talk about them. There is an endless parade of tools aiming to solve this problem of similarity-based annotation. Some of the ones you've had described are AAT. I guess PASA sort of counts in that it's using RNA-seq mapping to an assembled genome, repeat masker. The ones that I imagine most of you will have heard of a tool called BLAST, which is a very common sequence database search tool, sequence similarity detector, and it's been the workhorse of genomics for close to 40 years at this point. Another tool, Hammer, provides much higher sensitivity, ability to sort of look further back in time, handle a larger number of mutations without losing the signal of similarity at about the same speed for proteins and a little slower for DNA. This is software that my group contributes to on a pretty regular basis. A newer competitor to the field, MMSeq2, is a lot faster and about as sensitive as BLAST. There are an endless number of other tools that are in this kind of space, and if I try to put them all on the list, I'd have to use several slides. Repeat masker itself makes use of either cross-match or RM BLAST-N, which is a BLAST variant, or NHammer, which I developed. This slide is mostly just intended to point out that not only, right, are these sequence database search tools, these similarity-based tools, used for protein identification. I don't really have them listed here, but it's also used for RNA labeling and can be used for these transposable elements. And these are this is similarity-based approach using this database DFAM that we helped develop. And not sure why I've got those there. This is not similarity-based approach. This is model-based approach using TRF or Ultra, which I'll mention in just a moment. Okay, so I've already shown a picture of some pre-computed annotations, right, in the Ucsc genome browser. Let me show you a little bit more about them. These are the big names in the field: Ucsc genome browser, Ensemble genome browser. There are a variety of others. NCBI has their own, and there are many other browsers that contain a few other browsers that contain a large number of species and their annotations. There are lots of organism-specific genome browsers where they maybe bring in particular kinds of annotations or data sets that are really important to those specific organisms or organismal clades. I'm not going to list them here. I've got a little movie here of interacting with the Ucsc genome browser. This is the same region I had before, where we've got a little under a megabase of sequence for chromosome 4 in the human genome. This is showing that not only do you have a single gene annotation, but you might have proteoforms or isoforms of those where there's alternative splicing products. And we can look at the conservation, comparing the human genome against 100 other vertebrates. And there are some regions that are much more conserved than others, which suggests some measure of conservation. And that's all information that you can also use, right? That's sort of a model-based annotation to be able to label the genome in some way. And there are a bunch of things you can capture. including here, I've got now some methylation levels in particular parts of the genome, and that methylation is going to associate in some way with the activity level of particular genes. And now we're going to add another thing. This is adding in some single-cell RNA-seq data, which will show for a few of the genes. For each gene, this meta P1 clearly is some kind of a housekeeping gene, because we've got a whole bunch of different single-cell tissues, all with the same level of expression, whereas this alcohol dehydrogenase is much more expressed in some tissues and in, oh, that's funny. Here, in endothelial cells, it was not particularly well expressed. And so for any individual types, you have more or less expression, and that cell type expression can be pretty informative if you're trying to understand what's going on with a particular gene. We can make some more changes. Since we've got enough time, I'll let this video run. Here I'm going to add in the repeat masker viz track. And if I remember correctly with this video, it actually takes a second for it to, I don't know what happened, but it didn't. If you're far enough out, you can just see that repeats are it's chock full of repeats, and you can't really see much in the way of specific detail, but we'll get in closer in just a minute as we zoom into this genome. That's an important thing you really typically want to do. Right now we're at the level of seeing just a single gene, and this is me desperately trying to get it to show the visualization in the form that I want. And gosh, I think I'm going to need to zoom in again tenfold, and we'll finally have to reset it again, but we'll finally get to the point that we can see not just that there are particular kinds of repeats, but we can see individual repeats and how they exist in the context of each other, where in particular, like here's an L1P2 element that got inserted into this mere B4 element at some point in time. And so you can actually see the history of insertions that must have happened all that 150 million years ago, give or take, in this context. And Jack Roddy and my group built the interface for representing that on the Ucsc genome browser. Yeah, I think that's a great place to stop this video. It's not the only browser. Ensemble, which comes out of the European Molecular Biology Labs, that's the OMBL part of Ensemble, is another kind of a browser. And hey, they get more colors. Sometimes I really like to use it, but sometimes it seems to be overwhelmed and really quite slow. So I couldn't get a great video of this when I was trying all day yesterday. It kept being very slow to load, but it's got all the same sorts of information, just in a slightly different interface. You can see here all of the TEs again. I guess I'm TE obsessed in these video recordings. There's GC content, which just went away, right, with little squiggles to show part of the extent to which GC content rises and falls in different parts of the genome, isocores. And you can see the interface that's used here for adding or removing particular aspects of annotation that you might want to show. So there, that was me trying to give a little bit more sense of like how you might use existing tools to be able to engage with the annotation that's been done before you got to an organism, rather than just building your own annotation. Okay, so let's talk a tiny bit about how annotation actually works. Okay, I'm not going to get into too much detail, but I want to talk a little bit about it. So that was me pointing a laser in somebody's eyes. Really doesn't want to move forward. So model-based approaches, we kind of talked a little bit about this already, right? There's some model of what a thing looks like. And one example of that would be, for example, if you say, well, I know that tandem repeats, right, a sequence unit that gets repeated over and over again in tandem, like a tandem bicycle, that I know what they look like, which is that they're repetitive. Meaning if I know what's at one position in the sequence, and if I know that the repeat unit is length 4, so like it's an ATAC, 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 then I know that if I'm looking here, I should, on average, allowing for mutations, be able to look 4 positions back and find the same letter as I have right here. And if I'm in a region that's repetitive, that's going to happen a lot more than it would happen by chance. And so you can model that with some kind of a fancy probabilistic model. And Daniel Olson, who talked a couple days ago, has done so with a software tool called Ultra. And you can use a model like that just it creates probability models. And as long as you write it carefully, you can make it reasonably fast to do this kind of thing. And poof, you can recognize sequences that look a lot more repetitive than would happen by chance because you have a model and you compare it to a model of sequence that happens by chance and you just ask, is it more like this than you'd expect by chance? And in doing so, you can annotate a genome for, in this case, tannular repetitive sequence. That's an example of how you might do that, is you have to have some notion of what would it look like if it were this kind of thing versus if it were just random sequence. And then you can do essentially a likelihood ratio test of sorts to be able to decide if it's one thing versus another. But let's talk more about similarity-based approaches. So there are other sequence alignment-free approaches that get used sometimes for similarity-based approaches, but the vast majority of similarity-based annotation is sequence alignment-based. So I want to talk a little bit about what I mean by sequence alignment. Okay, so I've got a sequence. I'm going to call this the target. Okay, it's a bunch of letters. Here it's a DNA sequence, CC, AA, G, etc. Anybody know what that is? I think it's a bunch of letters I typed randomly, so it probably is nothing. It's been a long time since I made that slide. So, but it's something, right? And suppose then that I want to compare it to some other sequence. I'm going to call that the query. So I've got a query and a target. And I think, and I wonder, right, how similar are these two sequences to each other? Well, I could do things like I could count the number of C's, the number of A's, and the number of G's between the two and have some kind of number about that, but that wouldn't be that exciting. I could ask whether there are similar numbers of some kind of k-mer, where k is a length. So a 3-mer would be a length of a string of length 3, right? And I could ask whether they have the same kind of k-mer counts. And those would be alignment-free approaches. Or I could just do a thing called sequence alignment, where I actually do push this button. I do a thing that involves aligning those sequences, and poof, I've got an alignment. I imagine most of you have seen sequence alignments, and this is what they look like, right? I get each character from one sequence is aligned to each character from another sequence, except where they're not. Okay? And they're not always identical, and we'll talk a little bit more about that in just a second, but this is a sequence alignment. And if I can produce a sequence alignment, that can give me some information about whether and to what extent one sequence is similar to another sequence. So sequence alignments can be represented as paths in a two-dimensional matrix. Jeremy showed a dot plot of two chromosomes to each other, where if they were identical to each other, they would go from bottom left to top right. I tend to draw them going from top left to bottom right. It doesn't matter, but there's a path that you're taking from one place to another place in a matrix. And in a matrix representation like this, where a path corresponds to an alignment, the idea is that diagonal runs in a sequence alignment matrix like this correspond to a run of characters in one of the sequences, called the query, being aligned without any gaps, no insertions or deletions, to a run of characters in the other sequence. And I can see the blocks that I've presented here, whereas a vertical run of positions in that path would correspond, in this case, for example, to an insertion relative to the query, meaning there are characters that are present in the target that are not present in the query, and a horizontal run is a deletion relative to the query, for example, where there's stuff in the query that isn't found in the target. Right? And the reason that they're moving forward is it sort of used up characters in the query without using up any characters in the target. Okay, so there's a path in a matrix, and so you'll often see sequence alignment representation. Usually, I mean, this is how you usually see the sequence alignment, but you'll often see them represented as paths in the matrix, and that's what those directions mean. And being aligned or part of a diagonal doesn't necessarily mean that the characters are identical to each other. You can have substitutions. And the fact of aligning them is typically a sequence alignment tool's way of saying, yeah, I think that the ancestral sequence that these two were derived from, if it's working under the assumption since it's working under the assumption that they're related to each other, the ancestral character probably was one of those two. I don't know which one if I'm just looking at two sequences. And that there was some mutation, a substitution mutation, that changed one of them, but the other one is still the original one. Okay, so maybe T was ancestral and then a mutation to C occurred. And so they are, by being aligned, there is this presumption that these are homologous positions. They're positions that are derived from the same ancestral state, but that some mutations have occurred. And where there's no mutations, I'm drawing a vertical line here. That just suggests that nothing interesting happened, most likely, as opposed to the insertions and deletions where it's pretty obvious what is presumed to have happened. I can't see my notes, so I think I had a thing I wanted to say. Oh, I know what I wanted to say here. This is DNA. You do sequence alignments with protein also, right? So here's what a protein alignment might look like if it came out of the Hammer web server, where you've got one sequence that's your query, now made up of a 20-letter alphabet for amino acids, and another sequence that's your target. And where here I drew a vertical line if the characters were identical, here I draw a character if their characters are identical, or a plus sign if they're not identical, but not particularly penalized, like it's not such a big deal to change from an isoleucine to a valine. And then I'm missing those if it's likely to be kind of a not a preferred sort of a substitution mutation. And another thing that you get out of this top alignment, which is a pretty important observation, is that here, this is the first character of my query sequence. This is the 77th character of my query sequence. You can see that from the numbers. This is the 203rd character of my target sequence and the 281st. And it's a little bit strange, right? I didn't use all of whatever the target sequence was in this top one, right? Even though in this DNA alignment I showed you earlier, I used all of the characters, and that just sort of, it's used to highlight another thing that people talk about when they talk about sequence alignment, which is this difference between something called global alignment versus local alignment. And let me just use these examples to exemplify what I mean. Oh, sorry, global alignment is here, right? This is, global alignment is saying, I'm gonna, global, it's like all of, right? So the global alignment is a strategy where the alignment down here includes the full length of the query and the full length of the target, and all of the characters are used or represented in the sequence alignment, which is sometimes necessary if you're aligning a bunch of sequences where you know you've got the full gene of one and the full gene of the other, and you really want to see all of the characters aligned. But typically when you're performing annotation, you fall back to, this is what Blast and Hammer do, for example, you fall back to something called local alignment, which says it might be that only part of this sequence is related to part of this sequence, and the other parts are totally unrelated, and there's no reason to try to align the unrelated stuff. They're not related. And so you just produce an alignment of the parts that are related to each other, and so you end up with something like this, where not all of the characters from the original sequence are represented. And you can certainly see, like, if I'm trying to align a particular DNA element to a chromosome, that I'm really not going to be interested in that alignment having 50 million characters in that direction and 50 million characters in that direction and the few that I align in the middle. I want to just have the local part of the chromosome that matches up to the particular thing that I'm aligning to it. So that local alignment forms the basis of most of sequence annotation. Switching gears a tiny bit, what kinds of sequences do we do sequence alignment with? Well, you might have a protein sequence in hand. If you've got a protein sequence in hand, you're going to want to compare it to other protein sequences, most likely. And so you search it against a database of proteins. That's called protein-protein search. BLAST-P does that. P-Hammer does that. Each of the alignment tools will have some kind of a P variant, a protein variant that does protein-to-protein search. if you've got chromosomal DNA in hand, right, you have to remember, right, you got 2 strands, and your genes of interest might be on the top strand or might be on the bottom strand, where top strand tends to be the one, the terminology just means when you look at the genome that's captured in some, in the FASTA file that represents that, they don't show both strands, they just show one, because you can figure out what the other strand is from the first, just a reverse complement. So you have to search for things on both of those strands, the actual sequence you're given, and then reverse complement it and search in the other direction as well. Just a quick observation. So if you're working with those nucleotide sequences and you've got a nucleotide sequence in hand and you want to search it against other nucleotide sequences, like against transposable elements that are known in some library, like the library that's used inside of Repeat Massacre, I guess I've got this listed here, you're going to perform something called DNA search. Not always necessarily DNA, it could be RNA, but I think we all know how to turn a U into a T, right? So it's DNA search, where the characters are being compared at the level of nucleotide to nucleotide rather than thinking about amino acids. And that can be done either for RNA molecules. If you've got a database of RNA sequences, you can compare them at the level of the nucleotides or transposable elements. But if you've got protein coding DNA, right? Yeah, you've got DNA, it's in hand, and you could do a DNA to DNA search, but it turns out you're much more likely to do a better job of recognizing remote relationships that are due to some number of mutations by thinking of them at the level of the protein, even though all you have maybe in your hand initially is DNA. And that's because DNA comparison, every mutation seems like it matters, but at the protein level, that wobble base in the translation table frequently is irrelevant in terms of what the resulting amino acid is, and so you'd like to count those for free. And even changes that involve a couple of nucleotide changes might lead to amino acids that aren't a big deal to change. From an isoleucine to a leucine, it can have a little bit of a change, but frequently is irrelevant from the perspective of the resulting protein. And so you want to think about things at the protein level. You can do a much better job of looking deep back in time if you do so. And their strategy, as is typically used, is to find open reading frames with some kind of a length bound, and maybe use a gene finder that uses some models to predict what those are, translate those to the encoded amino acids, and then do a protein-protein search after that. You might also say, well, maybe there are problems with doing that. And I'd like to just, maybe I won't accurately, via my gene finding approach, identify all of the places where there's a protein coding gene, and I might miss some stuff. And so why don't I just take the protein and compare it directly to the DNA and just have the program internally say, oh, it's an amino acid here and it's a codon there, right? And just consider it in all 3 frames on one strand and then all 3 frames in the other strand. And so you get 6-frame translation for this thing called translated search, which is great. Also, if you're doing things like trying to account for exons and being able to consider the splice boundaries, or handling things like frame shift, inducing insertions or deletions in protein coding regions, which you might get from sequencing error or from neutral mutations in pseudogenized chunks of sequence, for example. And TFSY and LAST and Framer out of my group all do that quite well. Some better than others. I'll let you guess which one I think works best. A few more slides on this. So I've given you a sense that you can produce somehow a sequence alignment, and that sequence alignment corresponds to some path in this matrix. But at the heart of the process of producing this is the question, what's the right path? Which is to say, what's the correct alignment to use, right? I could name any alignment and it would be a path in that matrix, but which one is the right path to use? And that's dependent on a lot of things. There's a pretty slick algorithm that's been developed. for the computer science aware crowd in the room, right? It's a dynamic programming approach. It's called Smith-Waterman for finding high scoring alignments. There are even more advanced approaches that are related to that. I'm not going to talk about the details about how that algorithm works, but if you're interested, grab me afterwards or off in the genomics subgroup, and I'm happy to talk about the details of that. All of it boils down to having some notion of how much reward you should get for sticking two letters together if they are supportive of relationship between the sequences, right? Like sticking a letter to itself seems like a good idea. So if a what did I have here? If a leucine in one sequence is aligned to a leucine in the other sequence, then let's reward that with a score of 4, which is some magical number that was derived in some magical way that isn't actually magic. But what if I try to align a leucine to a tryptophan? Well, that should be penalized because that's likely to, first of all, it's likely to screw things up. Second of all, evolution has demonstrated that it doesn't tend to work all that well. And so it happens less often than what you would expect by chance. And so you get some kind of a negative score. And these matrices, are, once upon a time, people would hand build matrices based on their sense of how good or bad a particular change would be. We can now use a lot of data that we have from the results of evolution. Evolution tells us some things are more or less common than you expect by chance, and how much more or less influences the construction of matrices like this one. This is a Blossom 62 matrix, which is one, I would guess, got to be the most commonly used matrix for this sort of thing. There are others as well. There's a lot of nuance into how these scoring schemes are produced that I'm happy to talk about downstream, but rewards for aligning characters that are frequently accepted by evolution, penalties for things that aren't, penalties for insertions or deletions to alignments, and the scores of those influence the choice of what is the correct alignment of two sequences. And now it's worth thinking, I think, just a moment about how hard this problem is, computationally hard this problem is. If you've got some notion of scoring that you've already worked out, and you've got some notion of the best way to be able to find a high scoring alignment between two sequences, or the highest scoring, how long does that take to do? And if you've got a couple of sequences, let's say they're both the same length, which is n, then you have to fill in this entire matrix if you want to do it right for sure. And that means you've got an n squared matrix. If you're aligning two sequences that are linked 300, that's a few thousand cells, which is not very many. If you've got two sequences that are like long line 1 transposable elements, they're 10,000 nucleotides each, 10,000 times 10,000, that's 100 million. That got bigger in a hurry, and there's a bunch of calculations that have to be performed in each cell, so you're talking about a few billion calculations. It's actually not that big a deal still, because your computer, you know what your CPU speed is, it's like 3-something gigahertz most likely. That's 3 billion cycles per second. That's how many instructions it can perform. So a few billion instructions is no big deal. That's like a second. Okay, so cool, you're good. Except you don't just do one alignment, you do a lot of alignments. And if you're searching, for example, with proteins against UniProt-KB, which is a database of well-annotated, trusted protein sequences, you're talking about 500,000 or so sequences. If you go against a much larger data set from the UniProt data set, Tremble, it's at the European Molecular Biology Labs also, so Tremble. Now you're talking about 250 million sequences. 250 million seconds becomes a problem, right? And it gets worse. If you're looking at nucleotide sequences against the sequence read archive, you're looking at something on the order of 1 trillion sequences that are now present in that data set, and it's just getting bigger. So you need methods that don't fill in that whole matrix for every one of those sequences, or you will never be able to solve your problem. And that really drives everything that's at the forefront of modern sequence annotation, sequence alignment approaches, is desperately trying to not do this, right? What are the strategies you can use to make sure you still find the things you want to find, but avoid looking at the vast majority of those data sets? And if you think that sounds cool, then come and talk to me and I can discuss methods that are used to pull that off. But we're not going to get into the details of that now. Same problem exists if you're searching against genomes. You've got 3 billion base pairs. That's a lot of sequence to have to compare. And if you're comparing against a bunch of genomes, it's a bigger problem than that. Okay, so we want to do it fast. We have some way of building alignments, and there are a lot of ways to really speed things up. But one thing we really care about is, great, I get an alignment. An alignment, you add a bunch of character values up. They give you some kind of a score for that alignment. The score ends up being a function based on how the scoring matrices are identified. They're a function of how likely it is that the query sequence and the target sequence are descended from the same common ancestor, as opposed to just being unrelated. Low scores are easy to get by chance. You can just throw random sequences and you'll get low positive scores, and it's not a surprise at all. High scores are unlikely to happen by chance. If you get really high scores, then that's a pretty strong signal that these sequences are related to each other. But what do I mean by low and high and very high? Right? And it turns out that there's a lot of fancy math that lets us actually assign expectations to particular observed scores. We can identify what amount of p-values for those, right? What's the chance that a score this high or higher would occur by chance? And we can use those to compute this thing called an e-value, which I think I've got a slide for that, right? Amounts to this question. With this query sequence in hand, searching against that database of whatever its size is, if they were totally random, they were unrelated, right? How many alignments would I expect to see by chance? Right? That's the E, the expected number of alignments that I would expect to see by chance with this score S or higher. In other words, how many of those are just achieved by dumb luck? And so examples, if I see an E value of 10, that means I expected by chance, having nothing to do with relationship, an actual relationship, to see 10 sequences with a score that's that high, which means if I ran my software and I got 14 matches with an E value of 10, I expect 10 of those to have arisen by chance. So I don't really get excited about any of those 14, most likely. Maybe 4 of them are real. The e-values are imperfect estimates, so it might be that all 14 of them are not real. An e-value of 10 is not a reason to jump up and down and get excited about the thing you're looking at. An e-value of 1 means I expected to see one of those by chance. So if I get a hit back and it's got an e-value of 1, I'm not surprised. Right? E-value of 0.1 means I only expected to see 1/10 of a match with that highest score. I don't know how you get 1/10 of a match back, but you can think of that as meaning I would have needed to do searches on a database that was 10 times this size in order to expect to see 1. So I had just a 1 in 10 chance of seeing something like this randomly, which starts to feel like maybe that's pretty good. And if you like the p value of 0.05, then you might think p-value 0.05 is a good place to go. But just know there's a lot of noise in these e-value calculations. So I don't know that I'd get that excited about that either. 10 to the minus 6 means I had a 1 in a million chance of seeing a match with this good a score or better. And that's frequently kind of a reasonable place to actually, depending on your database size, to actually set up a threshold for, do I probably believe it or don't I? And there's still a lot of wiggle room around exactly what that e-value cutoff ought to be. Happy to talk more about it offline. And simple repeats in particular cause massive problems with predictions of those e-values. And so that's one of the reasons we hedge when we think about what e-values are actually meaningful. So in practice, there are, I mentioned, a lot of tools that are used for this: BLAST and Hammer, MMSeeks, if you want more sensitive or if you want faster. There are a variety of other tools that do that job as well that I'm not listing here. If I'm dealing with RNA that folds up into some structure, and if I happen to know the structure of some query elements, I can use a tool called Infernal that will not just look at the sequence, but it'll actually look at the likely structure that could be imposed by that sequence. There are a bunch of higher order tools that sort of wrap around these tools and others. Repeat Massacre is one of those. Bacta and DRAM are good for bacterial annotation. There's an endless list of those. If you're going to run them yourself, there are command line versions of all these tools. You can just download BLAST and run it on your own computer or on your own cluster. You can run Hammer. This is an example of the output of running Hammer on one of the sequences that's in its tutorial. You can also run them on the web, I think, if I push this. Maybe you have caught Giardia. This is a test case. You caught Giardia, your tummy feels terrible, and other people around you don't want to be around you anymore. And you think to yourself, maybe Giardia doesn't have an alcohol dehydrogenase gene, and if so, then I could drink it out of my system. So you grab the alcohol dehydrogenase sequence, and you pull up you focus your search for Fornicata, which is a clade that includes Giardia, and it also will speed up our search, and you click the button, and you get back a result. You wait a few more seconds than this, but I skipped 33 seconds, and then you see that, well, there's 3 matches in Fornicata species that have a 10 to the -6 kind of match. They're only 23, 25% identical, and if I look closely, they're dehydrogenases. There's an alcohol dehydrogenase, and it's in Giardia. You can still try. And the worst case that will happen is that you'll be really drunk while you have Giardia, and that'll be okay too. But maybe that's not the strategy you should use. And BLAST just told you that there are better ways to deal with Giardia than drinking yourself into a stupor. I'm sorry. I hope that was helpful. So that's the BLAST website. The Hammer website, the MMSeq's website is currently down because they're hosting a different thing on it, which suggests that they probably weren't getting all that much action on it. A lot of these have a thing called a RESTful interface, which allows you to write a script in Python or Bash or whatever language you like to work in that will send queries to the web server, and the web server will kick off results and eventually give them back to you or give you a link that you can look back up again. You can programmatically do all that on your computer, not on the browser, and do large-scale searches against the Hammer server or the BLAST server, which is a pretty nice interface to be able to use. This is my last slide. I just want to say that I'm happy to talk about what I've been talking about and extensions of those topics. The algorithm for finding optimal scoring alignment, talking more about where those scores come from, how those scores are translated into e-values, how do search tools avoid doing all of that work that I just complained about, how do annotation methods deal with possible multiple matches, how do they adjudicate between competitor annotations, how do you deal with sequencing error and tandem repeats and a variety of other things. And that is a place where I stop talking unless you've got questions.